Assignment 2.2, Part 2, by Samuel Marsh. When the anaerobic energy system creates two phosphate moles, we have a suitable and sufficient energy supply for the pyruvic acid. Now, instead of it being converted into lactic acid, going through a link reaction, it is catalyzed by the enzyme coenzyme A, forming the enzyme acetyl-CoA. This process allows our movements to be affected by the powerhouse of the cell, otherwise known as the mitochondria. The mitochondria, according to Apama V. Vidyaskar on the website Life Science, published on April 30th, are specialized structures serving as batteries, powering various functions of the cells and the organism as a whole, and are where aerobic respiration takes place. The second stage is the Krebs cycle. The acetyl-CoA produced from the first stage reacts with the oxaloacetic acid to form citric acid. This citric acid is oxidized through the Krebs cycle, a process and cycle for reactions. The hydrogens that are produced from the Krebs cycle are carried along by the electron transport chain in the mitochondria NAD and FAD, which are hydrogen carriers. Hydrogen ions are oxidized due to them losing an electron as they travel in pairs, meaning NAD and FAD. They transform into NADH and FADH. The hydrogen ions are moved around in pairs, causing a resynthesis of 30 moles of ATP, meaning it is the most efficient resynthesizing process. They are carried by FAD, meaning they get a yield themselves. The hydrogen electrons are carried by the electron transport chain. It supplies us with enough energy to resynthesize another extra 4 ATP, allowing an extra 34 ATP for the whole ATP resynthesis. The aerobic energy system creates large stores available for glycogen, meaning that the body should have enough food to be broken down when necessary. In addition to this, it provides low to moderate intensity exercise, meaning that the events where this system is used can last for long periods. As a result from this chain, byproducts are produced. However, they are only carbon dioxide and hydrogen, which are non-fatiguing products, meaning they can easily be removed for expiration. However, as the process is very long, the ATP resynthesis will be very slow, hence why the process can only occur in low-intensity work, where oxygen can be supplied, such as our experiment of the 20 minutes on the bike or marathon running. Muscle spindles, according to the Velocity PT Academy website, published on the 9th of November 2016, are stretch receptors, otherwise known as proprioceptors, that are widely dispersed throughout most skeletal muscles. They detect muscle movement and stretch, but also cause reflex that resist the stretch so it doesn't overextend. For example, once a contraction event has occurred, the muscle spindles detect the change in muscle length and send information to the spinal cord in the vertebrae. In addition, as this occurs, the secondary sensors detect the extent of the stretch, how large or small it is, and send this information to the overall central nervous system in the brain and the spinal cord. From here, it follows the common neuromuscular response, in which the central nervous system relays the information to the motor neurons to reduce the stretch. In sport, this can be exampled in weightlifting. The muscle spindles will increase their detection levels about the initial increase in movement, sending action potentials to the central nervous system about the intensity of the exercise and how many motor units are required in order to complete the movement. The higher the stretch detected, the higher the motor units are recruited. It informs how many motor units are needed to complete the action precisely, as if they didn't limit it, the muscle fibers and motor units will overwork and become strained and pulled. They send the impulse to the CNS and therefore the motor unit, ordering them to contract all together and cause the movement to occur. On the right here you can see a picture of the anatomy of a muscle spindle. The Golgi tendon organ, otherwise known as GTO, according to the Fundamental of Hand Therapy in 2007, are proprioceptors located in the tendon adjacent to the myotendinous junction. They detect changes in muscle tension and send this information to the central nervous system. They have nerve fibers that wrap around the end of them, attaching to the skeletal muscle fibers in the bone via the tendinous area. Steady-state exercise will increase the use of GTO, as when exercise increases, the GTO is stimulated by either an increase in stretch or contraction of the muscle, causing a myotatic reflex to occur. This means the working muscles won't overtense and, potentially injure the performer or create too high of an intensity of a steady state exercise, ruining the experiment and forcing the performer into anaerobic energy system, producing lactic acid. So, types of hypothesis. A null hypothesis, according to Martin Shuttleworth, published on February 3rd, 2008, on explorable.com, it suggests that a hypothesis which is null is one that the researcher tries to disprove, reject, or nullify. A single-tail hypothesis, according to Serbies on January 11th, 2017, on keydifferences.com, states that it is a test of null hypothesis, in which the alternative hypothesis is articulated directionally. A double-tailed hypothesis, according to Serbies again, on January 11th, 2017, on keydifferences.com, is an exhibited directionally of one of the null hypotheses, wherein the critical region is one or both the tails. So, the method for our experiment. Prior to the exercise, we constructed a warm-up, which involved the client cycling for 2 minutes in order to reduce the chance of injury or strain to the muscles whilst completing the event. Once the client had completed their warm-up, we took the readings of oxygen saturation in the blood pressure, heart rate, and breathing rate to make the results have a higher degree of accuracy once we had plotted them on the graph and once they had started exercise, and also so we could find the difference between the results and see how much they have changed from exercise to actually doing the exercise. Once we had taken all of their measurements, the client began their steady set exercise. In order to do this, we made them do 20 minutes of cycling at the aerobic training zone of 60 to 80% of their maximum heart rate. The maximum heart rate is calculated by 220 minus the age of the client. In our case, the client's maximum heart rate would be 2003 as they're 17. With the aerobic training zone being 121.8 to 162.4, once they entered this zone, we started to take their results. The timing were: heart rate, we did it prior, every minute, and post-exercise. Blood pressure, we did it prior, 10 minutes out of the 20, and post-exercise. For oxygen saturation, we did it prior, 3 minutes, 8 minutes, 13 minutes, and 18 minutes in, and post. For the lactate test, we did it 3 and 18 minutes in. For the Borg scale, otherwise known as the perceived intensity scale, every 2 minutes and post 2 times. And then for breathing rate, we did it prior, every 4 minutes, and post-exercise. Once the clients had finished their exercise, we immediately took their readings, same as stated before, as well as taking them 1 minute and 2 minutes after the exercise. This was so we can compare each individual's response to steady state exercise, prior, during, and after it, figuring out why the results occurred and if they match normative data, and see if any experimental methods occurred. So some of the equipment we used was stopwatch, a bike with some weights on the end of it to match the performer's weight so they can exercise efficiently, the Borg scale, the oxidation finger clip which measures the heart rate and the oxygen saturation, a lactate machine, and a blood pressure monitor. So here are the results of training and the raw data analysis. So for heart rate, the review. The average heart rate pre-exercise was 95.7 beats per minute. At 3 minutes of exercise, the average increased to 158 beats per minute, a massive increase of 60%. At 5 minutes, the average heart rate was 161, a minor increase of 2.5% from the 3 minute mark, showing a plateau. On the left here, you can see the graph of our free performer's heart rate. On the left is the beats per minute, and on the bottom is the timings. Before the exercise, we predicted that heart rate would increase during steady state exercise, but begin to plateau. This is due to the working muscles not needing an increase in oxygen as the intensity does not continue to rise as this aerobic exercise. The oxygen they're receiving in the blood from the heart is sufficient, meaning the heart rate doesn't need to increase. In order to measure the heart rate during our experiment, we used a device that was clipped onto the client's index finger. We used this device prior to the exercise, during it every minute, and post-exercise to find out and record the heart rate of our clients. All 3 of our clients experienced an anticipatory rise. The average heart rate before the experiment was 95.7 beats per minute, which is higher than the normative data. This is stated from Edward R. Laskowski, MD, on the Mayo Clinic, who states that the average is around 60 beats per minute. This anticipatory rise occurs due to the emotional release of noradrenaline, which is controlled by the hypothalamus. The sensory neurons inside of our body detect an increase of the noradrenaline and sends an impulse to the central nervous system, secreting the noradrenaline into the bloodstream, increasing the beat of the SA node and therefore the heart in preparation of the exercise. In addition to this, when the client hit 3 minutes to 18 minutes of their exercise, the heart rate only increased by 60% to 158. This is due to the heart rate furtherly increasing to supply the working muscles with enough oxygen so that they continue to contract. As the intensity doesn't increase as its aerobic exercise, the muscles do not require a constant increase in oxygen to work as they can meet the demand of the environment. The heart rate plateaus. This supports our hypothesis that there would be an increase in heart rate, then it would plateau from 3 to 18 minutes, or 3 to 20 minutes. The blood return to the heart will increase due to the venous return mechanisms and vascular shunt being more effective, forcing more blood back to the heart to pick up the oxygen. In addition, the precapillary sphincters are stimulated at the non-essential organs, allowing more blood to be sent to the working muscles for aerobic respiration and contraction. This reduces the blood being sent to an area, meaning the heart has less work to do and can continue to send the blood for a longer period over the duration of the aerobic event. This blood is being reduced to the area due to the precapillary sphincters undergoing vasoconstriction, or the shutting of blood vessels. Whilst 2 of our clients, Evelyn and Oshin, had the suggested normative data, Morgan's results didn't quite follow it. During steady set exercise, up until minute 6, her heart rate continued to increase. This may have been due to an experimental method, which is manipulating one variable to determine if changes in one variable cause changes in another variable, according to Verywell Mind. This may be due to Morgan working in her anaerobic energy system, rather than aerobic. Maximal heart rate is calculated by subtracting your age from 220, according to the Mayo Health Clinic. This means that for Morgan, it is 203, so she should be working in her aerobic training zone of 60 to 80% of the MHR, which is 142.1 and 162.4. At the 6th minute, her heart rate is 183. In an attempt to keep herself in this zone, Morgan may have carried on to work at an increased level, meaning her heart rate had to pump more oxygen to her working muscles over and over so they could contract, thus increasing Morgan's heart rate before she realised she was working too hard, causing a sudden drop in her heart rate readings. In our hypothesis, we also stated that the heart rate would drop rapidly back to what it was before, due to there being no lack of oxygen, reducing oxygen jet as oxygen was being consumed throughout the event. Whilst 2 of our results compared with the normative data, the more fit you are, the faster the recovery. Your heart rate drops most sharply in the first minute after you stop exercising. It should then fall to about 20 beats per minute by Berkeley-Well Wellness. Morgan's did not drop as sharply. From the minute 20 heart rate reading and then post-exercise reading, the drop was only a mere 2. This suggests that Morgan had been working in the anaerobic system in the event, which reduces lactic acid as a byproduct when no oxygen is present. We can back this up by using her lactate energy system graph, which is going to be shown later on, in which lactate levels spiked at minute 8, suggesting she was working at a way too high intensity in the anaerobic training zone, meaning that the cool down for her would have taken longer compared to the other 2 clients.